Okay. Very good evening, everyone. We are going to understand today about the microscope. See, name itself says microscope. So I'm writing here microscope. Microscope. See, we are using telescope also, right? So tele means long distance and scope means to see. That means the instrument that is used to observe the object which are at a very long distance are called as telescope, right? Likewise, here in name itself says micro and a scope. So scope means again to see, okay, or to observe, you can say. Okay, and micro means very tiny objects. Very tiny means which cannot be seen with our naked eyes, which cannot be observed with our naked eyes. Naked eyes, right, everyone? Okay. So see, in meaning itself, we have the definition. Okay. So microscope is what? It is the optical instrument. So I'm writing here definition of microscope. What is microscope? The optical instrument. Optical instrument. The optical instrument used to watch, okay, or not watch, we write observe, see or observe. Okay, the optical instrument used to see or observe very tiny particle, you can say, organism, you can say also. Okay, so here we are like discussing about organism. So I am writing here organism. So very tiny organism or objects also, you can write objects. Okay, which cannot be seen. Which cannot. Someone is in waiting room, beta. Wait a second. Okay. Okay. Which cannot be seen or observed with our naked eyes. With our naked eyes. Naked eyes. Very simple, right? That instrument is called as beta microscope. Microscope. Easy, what is microscope is? The optical instrument used to see or observe very tiny organism or object which cannot be seen with our naked eyes or observed with our naked eyes is called as microscope, right?
Okay, sir.
It's called as microscope. Sriram, are you there?
Yes, sir.
Okay. Any doubt here in microscope?
No, sir.
Okay. Now, generally in biology laboratory, yes, beta?
Sir, one new student came, sir.
Who?
So, don't know.
What's your name, beta?
Sir, ma'am, it's Purujit Raman.
Will you please repeat?
It's Purujit Raman.
Yeah, Purujit Raman. I am Hemant sir, beta. I am the science faculty, okay?
Okay, ma'am.
And I am Hemant sir. Okay?
Okay, ma'am.
I am Hemant sir, sir.
Okay, sir.
Okay. Okay, you are in 8th class, na? From Hyderabad only, right?
I'm not from Hyderabad. I'm currently studying in a Dubai school.
Oh, yeah. Okay. Yes. Okay, so you're from Dubai, means you stayed there. Okay, no problem. So we have started with a new chapter, which is microorganism, friend and foe. Okay, you must be having, right?
I didn't learn. I am not. I don't think I didn't learn this.
No, you must be having in your syllabus, na microorganism.
So,
It is 8th class syllabus, beta. Okay. Do you have your syllabus with you now?
yes, I do.
Okay, just do check.
Okay. Can I have a second for that?
Yeah. And everyone, start your cameras, beta. Keep your cameras on. Turn on your cameras, everyone.
Mom, I got the syllabus.
Okay, and just do check. You have microorganism.
Yes, ma'am. I do.
Okay. So that chapter we have started beta. First chapter we already covered. We have covered few chapters, but for you we can repeat that also. Don't worry. Okay. Sorry. So we have started microorganism, and this is our second class beta. And here we are discussing. We are repeating the microscope. Okay. So we just completed the definition. Microscope is nothing but the optical instrument. It is nothing but the optical instrument used to see or observe very tiny organism or object which cannot be seen with our naked eyes. Okay, that instrument is called as beta microscope. Okay, and meaning of microscope itself says its definition. So micro means very tiny, and very tiny means which cannot be observed or see with our naked eyes. Okay, and scope means to see or to observe it. Okay, that means it is an instrument which is used to observe very tiny particles which cannot be seen with our naked eyes, called as microscope. Okay, now we are going to discuss about structure of microscope. Structure. Okay, so structure we will discuss from the notes now. So I want just take a screenshot for that particular part.
Yes, sir.
Everyone. Okay, I'm zooming in now.
Okay, sir.
Okay.
Sir, did you share the textbook PDF of this lesson?
Yes, beta, on your WhatsApp group, I just shared like
Okay,
few
thank
minutes
you,
back.
sir.
Yeah. Okay. See, a microscope is an instrument. So it is an instrument that makes the use of lenses. So here we are using lenses, beta, and generally convex lens we are using. So this lens here, it is called as eyepiece, and here we have like different power of the lenses. Okay, which are called as objective lens. Okay, so make of the lenses to make the smaller object appear very large. Okay, so smaller object appears very large. Usually a compound microscope. See why we have used word compound here, because in microscope we are using two lenses together simultaneously. Okay, we are using two lenses simultaneously. That's why it is called as with combination. That's why it is called as compound microscope. Understood everyone why it is called as compound microscope?
Okay, sir.
Yes. It is used for viewing very tiny organisms. Okay, like amoeba, paramecium,
Yes,
euglena,
sir.
bacteria, okay, which cannot be seen with our naked eyes, but we can see them with the help of microscope. Okay, the different parts of the compound, okay, compound microscopes are shown here. So now we are going to discuss about different parts, means the structure of microscope. Okay, the object to be viewed under the microscope. So whatever the object, see how objects are placed. Okay, So we are having one glass sheet like this, small glass sheet. Okay. Here we are putting whatever the object that we want to see. Okay. And here we are covering with the cover slip, beta cover slip, in order to protect the lens of microscope. Which lens? The objective lens. Okay. So the part which is used, or the organism or any object which is used in order to see in a large form, is called as beta specimen. It's called as what? Specimen. Understood what a specimen is? Everyone?
Yes, sir.
So specimen
Yes,
is
sir.
what? It is the object, okay, which have or which can be like enlarged with the help of microscope, and we can observe them clearly. It's called as, or are called as specimen. A thin sheet of the glass, okay, called microscopic slide. So this is called as beta microscopic slide. Okay, is used to hold the small sample of the specimen. So here, this is the specimen. Okay, this is the specimen which we have to see. A second much thinner sheet of the glass. Okay, so see, first we have glass slide. Okay, then we are like keeping that specimen, and over it, okay, above it, we are using that glass.
Cover slip.
Yeah, and then very thin glass called cover slip. It's called as what? Cover slip. Okay. Squad cover slip is placed over the sample. Okay, so it is placed over the sample. Okay. The cover slip protects the microscopic objective lens by preventing it from coming into the contact with the specimen sample. Okay. So that cover slip, okay, here we have cover slip. It protects the lens of the objective lens or lens of the microscope, which protects it from touching that lens to the object, right? Is it everyone? So this is the use of cover slip. It also helps to create and even thinness of the sample. Okay, so it's really very thin. So whatever the thin object we have, they should not be going to break with the when objective lens is going to touch them. So it also protects them. Okay.
Sir, then what are the adjustment screws?
Beta.
Sir, then what are the force adjustment screw, fine adjustment?
Okay, these are in order to focus them clearly. Okay, sometimes see, object is not being clear, so it appears blur, beta, blur. So
Yes.
these adjustment screw we had to focus in order to form the. We have to focus in such a way that we will get the clear image of the object. Okay, so by which we can adjust the focal length. Okay, beta. Suppose if we are getting blur image, so we have to adjust with the help of screw. Okay, so that it may move up sometime or it may move down. Okay, in order to, so whenever during the movement, if we get the clear image, we have to stop moving that object. Okay.
Sir, then how does electron microscope work? Sir, like,
Beta.
then how does electron microscope work?
Electron microscope. Okay. It is like very complicated instrument, beta. It is very, in order to see electron, we had to first create the vacuum. Then whatever the object that we want to see, we had to like pressurize that object inside the vacuum chamber at a very, very, very low pressure. Okay. Then electrical discharge tube is being placed there, and on a fluorescent screen, that image is going to form of the electron. So that's why electronic level object can be seen with the help of electron microscope. Okay.
sir.
Yeah. I will show you how microscope is going to work and how we have to test this. Beta?
Okay, sir.
Okay. Okay, the basic concept to use compound microscope. Okay, basic steps whenever we are going to use. So first we have to clean the mirror. Okay, that mirror we have to clean first so that we will get clear image. Clear light. Okay. Then by using the soft and clean cloth, then place the slide. Okay, so this is the stage beta. This is called as what stage? Here we had to place the slide. Okay, and here we have like stopper. Okay, click so that it is not going to move. So it makes the slide fix at a particular place. Okay, place the slide with the specimen on the stage. Adjust the focus of the eyepiece and the objective lens with the help of your teacher. Means with the help of your like laboratory assistants or maybe teacher, here to like adjust with the help of that screw beta screw. Okay, they are adjusting screw. View the slide through the eyepiece. Okay, and we can see that of specimen clearly with the help of microscope. Okay, so now
Sir, iris. Sir, then where is iris located? Iris.
Iris is the part of our eye, beta.
No, sir. In the video that you showed last class, it was a part of the microscope.
Iris no, beta. It is eyepiece. This is the
Yes,
eyepiece.
sir.
Yeah, over here at the top. This is the eyepiece, beta. That one. At the top. See
So there was
why?
actually, actually an iris in the video. They showed iris.
Yes, sir.
Like it wasn't a natural iris, but it was made
Yeah,
of.
it's lens, beta. It's actually the lens, the glass lens.
Yeah.
Okay, sir.
Yeah. Okay. So it is used in order to see with the help of eye. That's why it is called as, that lens is called as eyepiece. And that lens near it is order to like grab the image of the objects. That's why it is called as objective lens. Okay.
Sir, the iris is between the objective lens and the pipe, right?
Iris, no, it's eyepiece, beta.
No, sir, there is actually
Yes, sir. It's there in
iris
the
which
video.
controls the amount of light passing through the eye lens.
Okay, okay. See, that iris is the part of our lens, and that we discussed yesterday in last class. Okay, see, this is our muscular lens. Okay, and here we have the our part of the eye. Here at the center we are having iris, and here we are having pupil. So that iris used to sometime contract or relax or dilate sometime. So that see, this is the pupil, and from here the light enter inside, and here we have beta retina. Okay, so how much amount of going to light enter inside the eye? This is controlled by iris. This is iris beta. Okay, this is iris. Let me show you. Let me show you how microscope works and how we can see that. Wait.
Okay.
It has only one eyepiece.
Okay, okay. See, as science and technology is being developing, it becomes more like advanced, you can say. So whatever the microscope that we have observed is advanced one, beta. Huh? Okay. And
Okay, sir.
generally, you must be having the mirror at the like base in order to source of light, right?
Yes.
Yes, but here it's like LED light source is being provided here. That's why we had to plug in. Okay, so now let's discuss about the types of microorganism. So microorganisms are only visible with the help of microscope. That's why they are called as microorganism. Okay, so organism which can be seen only with the help of microscope are called as microorganism. Easy or very easy? Yes, do reply. It's easy or very easy, beta?
It's very easy.
Very.
Yes, yes. So keep replying, no, in between. Okay. So micro means very small, very tiny. Okay. And they are very tiny so that we cannot be see with our naked eyes. That's why we to require microscope in order to see such organism, and they are called as microbes also. So microorganisms are the most abundant organism on our planet. You know, they present everywhere, in water, in soil, in food, in air, mountain, forest, like in ice cold region, at very high temperature, temperate region, everywhere they present better. Everywhere. Okay. Keep replying.
Okay, sir.
Okay. They are found almost everywhere. See, it is given here. They are found almost everywhere. Like in ice cold region to hot spring, and even in desert. Okay, even inside the body of the animal, plant, human being, etc. So they present everywhere, beta, everywhere. So they are Arush again entered in the class.
Region also,
Yes.
no?
Huh, beta?
In ice cold region also, they will
Yes, yes. They present everywhere, beta. Yes. Okay. Some live alone, so they are free living. Some live alone, so they are free living. Okay. While other grow in groups called colonies, so they are present in colonies also. Okay. They may be unicellular, means they are made up of single cell, or they may be multicellular, means they are made up of many cells. Okay, like a hydra. Okay, it is made up of many cells, and we are able to observe them with the help of microscope only.
Okay, sir.
Yes. Microorganism can be divided into 5 major groups. So there are 5 major group of microorganism. Okay, one is called bacteria, other is called protozoa, then we have fungi, then we have algae, and then we have viruses. We have viruses also. Understood? Bacteria, protozoa, fungi, algae, and viruses. Now let's discuss about first bacteria. So I won't just repeat that part. Just repeat it quickly.
Okay, sir. Repeating.
Yeah. Done.
No, sir. Done, sir.
Okay. Now let's discuss about the bacteria. Okay, see. Okay. So bacteria are among the smallest and oldest organism of our planet. Okay. See, there are many techniques by which the life or the age of any organism can be determined. Life means, like suppose we have dinosaurs better. We are having dinosaurs on earth, right? So we have their remnants. So on the basis of observation and a carbon dating technique, we are able to find the, when the last dinosaur got dead on earth's surface, right? And we know dinosaurs were about 6.7, 6.5 or maybe 7 crore year ago they used to live on earth, right? Dinosaurs, right, everyone?
Sir.
So yeah, so that age of the dinosaur, like we have calculated with the help of carbon dating. Yes, carbon dating technique. Okay, so that with the help of carbon dating technique, it has been proven that the bacteria are the oldest organism on our planet. Okay, oldest one. Bacteria are unicellular. हाँ बेटा.
Sir, can you say again?
See, bacteria are the oldest organism found on our planet called Earth. Okay, so when life came on Earth, so it came in the form of bacteria only first. Okay.
Okay, sir.
Yes. So bacteria are unicellular.
Okay,
Unicellular means they are made up of single cell. Yes. Single cell only. Yes. Different bacteria have different shapes. Okay. So different types of bacteria having different shape. Okay. So they may be rod shaped. If they are rod shaped, they are called as bacilli. So I'm writing here their name. If bacteria are rod shaped. Rod shaped. Okay, so if they are rod shaped, so they are called as bacilli. P-A-T-I-L-L-I. They are called as bacilli. Okay. If number one, number two. Okay. If they are spiral shaped, spiral. Okay. See here, they are spiral shaped. So they are called as spirilla. Spirilla. Okay. If they are comma shaped, beta, comma shaped.
So what, sir?
If they are comma shaped, comma. Okay.
Yes, sir.
Yeah. Comma shape like this.
Okay,
Like
sir.
this. Yeah.
So
So
I have a doubt.
हाँ बेटा.
So being in different shape, does it like specify them in a different way?
Yes. It specifies their types also. Okay.
Okay, sir.
Yeah. If they are comma shape, they are called as vibrio. Okay. If they are spherical shape, beta, spherical.
Okay,
They
sir.
are called as foci. They are called as foci. Sorry. Okay. So these are the 4 types of bacteria on the basis of shape. Okay, their shape. Okay. So if they are like rod shape, they are called as bacillus. If they are called as spherical, they are called as foci. If they are comma shape, they are called as vibrio. And if they are spiral shape, they are called as spirilla. Okay. So lactobacillus. See, lactobacillus. Again, here we have bacillus. That
Is
means
in
they
rod
are
shape.
rod
Rod
shape. Yes. And streptococcus. So here coccus we have
Is in
foci.
spherical shape.
So it is spherical in shape. Yes. Easy
Sir
or
will
very?
repeat this part. Sir will repeat
Okay.
this part.
Okay.
Sir,
Okay.
easy.
Okay. See. So bacteria are the oldest organism found on Earth. Means when life came on Earth, it came in the form of bacteria only. Okay. Now we have different types of bacteria, and their type is on the basis of their shape beta. If bacteria are rod shaped, they are called as bacilli. Okay, or bacillus. If they are spiral shape means like this, as you can see here. Okay, they are spiral shaped beta, that one. Okay, so if they are spiral shaped, they are called as what? Spirilla. If they are comma shaped like this, if they are comma shaped, they are called as trio. But if they are spherical in shape, then they are called as foci. Understood now?
Okay, sir.
Sir, we will repeat it, sir. We'll read it.
हाँ बेटा.
Sir will read it, sir.
Yeah, you can read it once.
Okay, sir.
Okay, so here fact file is given here. We have to go through that. A gram of the soil contain. Okay, a gram of soil contain about 40 million bacteria. Okay, so in 1 gram of soil, beta, 1 gram of soil. 1 gram of soil contain 40 million. 1 million means 10 lakh. Okay, so you can imagine 40 million.
40 million means
Yes.
4 crores.
40 million bacteria.
Means 4 crores of bacteria.
10 million. Yes. Right. See, it's very large number, no?
Yes, sir.
Yes. Have you repeated everyone?
Yes, sir.
Yes.
So I want just take a screenshot also. Some written work is also there.
Sir, I wrote it.
Okay. Okay, so this is all about bacteria. Now we will discuss about protozoa. Okay, so protozoa are nothing but the group of organism. Okay, or they are group of animal-like only. Okay, so they are group of animal-like organism, but they are unicellular. Okay, and sometimes they may cause disease to human being, even plant and other animals also. Okay, so they may cause diseases also. Okay, beta. We have example called amoeba, paramecium, and euglena. Okay. After euglena, yeah. Beta. Okay. These are the examples of protozoa. Example of protozoa. Okay. Let's discuss about now. Okay.
So,
Beta.
the Vedic class has started, sir.
It's already time done.
No, sir. 5 minutes remaining, but
Can we leave, sir?
Okay. Everyone having Vedic class?
Sir, some are there.
Sir, none class, sir.
means who has by this course of max and science both, they will get free.
Okay. So only new student have to stay. Otherwise, they may leave the class. So I think Pranav, Karthik, are you new, beta?
No, sir, they're old.
Yes, sir.
Okay. And Raman, no? Gurujit Raman, na?
Yes, sir. Yes, sir.
Okay. So Gurujit, have you understood the concept, beta?
Yes, sir.
Okay. And what about you, Pranav?
Yes, sir. I understood.
Okay. Any doubt here, beta?
No, sir.